Reports emerging from South America indicate a significant surge in rodent populations across various regions of Argentina, a biological phenomenon often linked to the transmission of hantavirus. This escalation in rodent activity, frequently referred to in ecological terms as a masting event or a rodent irruption, has placed public health surveillance systems on high alert. The concern stems from the well-documented role of specific rodent species as natural reservoirs for hantaviruses, which are a family of viruses primarily spread by rodents. While the headline suggests a global threat, the immediate focus remains on the localized ecological shifts in Argentina and the resulting risk to human populations in proximity to these rodent clusters. To understand the gravity of the situation, one must look at the nature of the virus itself. Hantaviruses are enveloped RNA viruses belonging to the family Bunyaviridae. Unlike many other viral threats that dominate news cycles, hantaviruses are not typically known for widespread human-to-human transmission, with one notable exception found in South America. The primary mode of transmission to humans occurs through the inhalation of aerosolized viral particles. These particles are shed in the saliva, urine, and feces of infected rodents. When these waste products dry and are disturbed, through activities such as sweeping, agricultural work, or even moving stored items in infested sheds, the virus becomes airborne. Once inhaled, the virus targets the endothelium, the thin layer of cells lining the blood vessels, leading to significant physiological distress. In the Americas, this typically manifests as Hantavirus Pulmonary Syndrome, a severe and often fatal respiratory disease. The current situation in Argentina is particularly scrutinized by the international scientific community because the specific strain prevalent in the southern Andean region, known as the Andes virus, has shown the unique capability for limited person-to-person transmission. This characteristic elevates the virus from a localized rural health concern to a matter of broader epidemiological surveillance. Public health officials are currently monitoring environmental factors that contribute to these rodent booms, such as unusual flowering cycles of local bamboo species or shifts in agricultural patterns, which provide an overabundance of food for the long-tailed pygmy rice rat and other known carriers. The geographic focus includes both rural agricultural zones and the peri-urban fringes where human habitation meets wilderness, creating a high-risk interface for cross-species transmission. As the rodent population density increases, the probability of human-rodent encounters rises proportionally, necessitating a robust and rapid response from provincial and national health authorities to prevent a localized outbreak from escalating into a broader public health crisis. Public health agencies and specialized laboratories in Argentina are currently mobilizing to address the increased risk posed by the reported rodent surge. The National Administration of Laboratories and Health Institutes, often central to such efforts, serves as the primary diagnostic hub for verifying suspected hantavirus cases. The diagnostic process is rigorous, involving molecular testing such as polymerase chain reaction, or PCR, to detect the presence of viral RNA in blood or tissue samples. Additionally, serological assays are employed to identify antibodies, specifically immunoglobulin M and G, which indicate a recent or past infection. These laboratory facilities are critical in distinguishing hantavirus symptoms, which initially mimic common influenza, from other regional endemic diseases like dengue or Argentinian hemorrhagic fever. Clinical presentation typically begins with a prodromal phase characterized by high fever, muscle aches, and fatigue. However, the rapid progression to the cardiopulmonary phase, where the lungs fill with fluid and blood pressure drops significantly, requires immediate intensive care intervention. Hospitals in the affected regions are reportedly reviewing their protocols for respiratory isolation and the management of acute respiratory distress syndrome, while the source material points toward a potential pandemic scenario. Medical experts emphasize that hantavirus outbreaks are historically self-limiting, provided that strict environmental and clinical controls are maintained. The focus for health officials is currently on active surveillance, the systematic collection and analysis of data regarding both human cases and rodent infection rates. Field teams are tasked with trapping rodents in high-risk areas to determine the prevalence of the virus within the local population. This data is essential for mapping hot zones, where the risk of transmission is highest. Regarding trade and logistics, while there are no official reports of shipping disruptions, general public health guidance suggests that warehouses and storage facilities in the affected provinces implement enhanced pest control measures. The concern in the international community remains focused on the Andes strain's capacity for interhuman spread, as documented in previous outbreaks in towns like Epuyén. Consequently, any confirmed case triggers an immediate epidemiological investigation to trace contacts and implement temporary isolation measures where necessary. At this stage, official statements from health ministries emphasize caution over alarm, focusing on the deployment of resources to rural clinics and the reinforcement of diagnostic capabilities in provincial labs to ensure that any potential jump from rodents to humans is detected and contained at the earliest possible moment. Prevention and public guidance remain the most effective tools in managing the risk of hantavirus during a rodent boom. Health authorities are issuing clear directives to residents and travellers in affected Argentine provinces to minimise the risk of exposure. The core of these recommendations is the disruption of the rodent-human interface. This includes securing homes and outbuildings by sealing any openings larger than a quarter inch, which serves as an entry point for small rodents. In areas where rodent activity is confirmed, the public is advised against dry sweeping or vacuuming spaces that may contain rodent droppings. Instead, a process of wet disinfection is recommended, using a mixture of water and bleach to saturate potentially contaminated surfaces before cleaning. This prevents the virus from becoming aerosolized and inhaled. Furthermore, individuals engaged in outdoor activities such as camping or trekking are urged to stay on marked trails and avoid sleeping directly on the ground in areas with high rodent density. The limits of current medical knowledge regarding hantavirus are an important factor in public health communication. Currently, there is no specific antiviral treatment or widely available vaccine for Hantavirus Pulmonary Syndrome. Medical management is supportive, focusing on oxygen therapy and maintaining fluid balance in an intensive care setting. The mortality rate for the disease remains high, often cited between 30 and 40%, which underscores the necessity of early detection and preventative behaviour. Surveillance systems are also looking at long-term ecological data to predict future eruptions. Yet the exact triggers for these massive rodent wars remain a subject of ongoing study, involving complex interactions between climate change, local flora, and predator-prey dynamics. As the situation in Argentina develops, the international community continues to monitor for any signs of geographic expansion of the virus. For now, the response is a balance of rigorous science and community education, aimed at containing the biological threat at its source. Honest reporting indicates that while the potential for a larger outbreak exists, the current emphasis is on localized containment and the protection of vulnerable populations through informed public health practices. Stay informed as we continue to track the developments of this ecological and health event. Subscribe for more in-depth reports on global health security. In response to the reported escalation of rodent activity in Argentina, the national and provincial health ministries have intensified their epidemiological vigil, focusing on the sophisticated laboratory and clinical networks required to manage orthohantavirus. At the center of this diagnostic effort is the National Administration of Laboratories and Health Institutes, known as ANLIS Malbrán, which operates as the primary reference center for infectious diseases in the region. The process of verifying a hantavirus infection is complex and requires specialized biosafety level 3 facilities to handle potentially lethal viral samples safely. When a patient presents at a rural clinic with the characteristic prodromal symptoms, typically high fever, severe myalgia, and gastrointestinal distress, health protocols mandate the immediate collection of blood samples for molecular and serological analysis. The primary diagnostic tool is the reverse transcription polymerase chain reaction, or RT-PCR, which allows scientists to detect the specific genetic sequence of the hantavirus RNA in the patient's blood even before the immune system has produced a measurable antibody response. This early detection is critical because the transition from initial flu-like symptoms to hantavirus pulmonary syndrome can occur within hours, leading to rapid capillary leak and pulmonary edema, while the sensationalist framing of a global pandemic is scrutinized by the scientific community. Virologists point to the unique characteristics of the Andes virus strain prevalent in southern Argentina as a legitimate reason for heightened surveillance. Unlike most other hantaviruses found globally, such as the sin nombre virus in North America, the Andes virus has a documented history of limited person-to-person transmission. This specific biological capability necessitates strict isolation protocols in hospitals and the rigorous tracing of all close contacts of a confirmed patient. In previous localized outbreaks, such as those seen in the Chubut province, health officials implemented mandatory domestic isolation for high-risk contacts to break the chain of transmission. Current clinical developments also focus on the readiness of intensive care units. Since there is no specific antiviral therapy proven to cure the infection, medical management relies heavily on early aggressive supportive care, including the use of mechanical ventilation and, in some advanced facilities, extracorporeal membrane oxygenation, or ECMO, to stabilize patients while their bodies fight the viral load. The impact of a rodent surge also extends into the logistics and trade sectors, particularly in agricultural zones where grain storage provides an ideal environment for rodent proliferation. Although there are no confirmed reports of international trade disruptions at this stage, standard public health guidelines for shipping and maritime transport require the implementation of stringent rodent control measures to prevent the accidental transport of infected hosts in cargo containers or grain silos. Regional health agencies have emphasized that the risk to the global community remains low as long as these zoonotic interfaces are managed and the virus does not undergo significant mutations that would facilitate more efficient human-to-human spread. Meanwhile, in the laboratories, researchers continue to study the viral load in captured specimens of the long-tailed pygmy rice rat, the primary reservoir host, to determine if the current population boom correlates with a higher-than-average viral prevalence within the rodent community. This data is essential for calibrating public health warnings and determining whether the current situation represents a standard seasonal spike or a more significant ecological anomaly. The disparity between alarming headlines and the measured response of health agencies highlights the challenge of communicating complex zoonotic risks. While the rodent irruption is a verified biological event requiring a robust medical response, the leap to a global pandemic remains a theoretical risk rather than a documented reality, contingent upon the continued effectiveness of local containment and the inherent biological limitations of the virus itself. Addressing the surge in rodent populations and the attendant risk of hantavirus transmission requires a rigorous commitment to both individual prevention and systemic public health surveillance. For residents and workers in the affected Argentine provinces, the front line of defense is the systematic elimination of rodent habitats and the strict adherence to decontamination protocols. Public health guidance emphasizes that prevention begins with structural integrity. Officials advise sealing all entry points in homes, granaries, and sheds with durable materials like steel wool or cement, as rodents can navigate openings as small as a quarter inch. Beyond exclusion, the management of the immediate environment is critical. This involves the removal of brush, wood piles, and trash from the perimeter of dwellings to eliminate nesting sites. In agricultural and rural settings, where the interface between humans and the long-tailed pygmy rice rat is most frequent, the emphasis is placed on the proper storage of food and animal feed in rodent-proof containers. These practical steps are not merely suggestions, but are essential components of a strategy to reduce the density of the viral reservoir in proximity to human activity. The specific mechanism of hantavirus transmission, aerosolization, dictates the precise nature of cleaning and disinfection protocols. Health authorities warn that traditional cleaning methods, such as sweeping or vacuuming, are strictly prohibited in potentially infested areas because these actions disturb dried rodent excreta, lofting viral particles into the air where they can be easily inhaled. Instead, the mandated procedure is wet disinfection. This involves thoroughly soaking any contaminated surfaces, droppings, or nesting materials with a solution consisting of 1 part household bleach to 9 parts water. After a contact time of at least 10 minutes, the material should be removed using gloves and disposed of in sealed plastic bags. This method is effective because hantaviruses are enveloped viruses, meaning their outer lipid layer is highly susceptible to common disinfectants. For those entering enclosed spaces that have been vacant for extended periods, such as seasonal cabins or storage sheds, the directive is to allow the space to ventilate for at least 30 minutes before entry, further reducing the concentration of any airborne pathogens on a broader scale. Public health surveillance in 2026 relies on an integrated approach that combines ecological monitoring with clinical reporting. Scientists are closely tracking the flowering cycles of native flora, such as the collyhue bamboo, which can trigger the masting events that provide the caloric surplus necessary for a rodent population explosion. By understanding these environmental precursors, authorities can issue preemptive warnings to specific regions before human cases begin to emerge. However, an honest assessment of current medical capabilities reveals significant limits. There remains no specific antiviral treatment or widely available vaccine for hantavirus. The ethical responsibility of health agencies, therefore, lies in the transparent communication of risk and the prioritization of early supportive care. The high mortality rate associated with Hantavirus Pulmonary Syndrome, often exceeding 30%, underscores the gravity of the situation and the necessity of rapid clinical response. The current rodent war in Argentina serves as a reminder of the delicate balance between human development and natural ecosystems. While the term pandemic is often used to describe any significant viral threat, the current hantavirus situation is more accurately categorized as a localized zoonotic crisis, with the potential for expansion if surveillance and containment efforts falter. The ethical imperative for the global health community is to support regional efforts in South America, recognizing that in an interconnected world, an outbreak in one corner of the globe is a matter of concern for all. The goal is to move from a reactive posture to one of proactive stewardship, utilizing the best available ecological and medical data to protect vulnerable populations. As we monitor the situation, the focus remains on calm, evidence-based action rather than speculation. Effective prevention and vigilant surveillance are the most powerful tools